So first, I would like to thank our organizers and NITMB for this fantastic opportunity. Today, I'm going to share with you a system in biology that I think is very interesting from my point of view. I have been starting to look at this system very recently. And unlike I said here, I did not take the lightning word very literally. So
there are many types of lightning.
True. Okay. So my system that I'm going to talk about is ferns. Ferns are vascular seed-free plants, and they grow out of spores. And they have a very long, interesting life cycle, as you can see here. We are very interested in the very early stages of this life cycle, mainly the one that's highlighted in a red box. So we're interested in seeing how the ferns would grow from a gametophyte that is very circular and rounded into something that's very like a heart shape. So you have a heart shape and there's a meristem notch at the center with a very dynamic cell division area. So our collaborators in plant pathology have been looking into this, and they were speculating that the formation of this meristem notch is actually motivated by some signal that's coming from the spore coat. So to be able to understand this process better, we tried to construct a toy PDE model where, for simplicity, we assume that you have two signals, one that's coming from the spore coat and another that's coming from the apical part or from the top part. And those two signals are going to overlap. And at the maximal intensity at the two sites, you will have potential sites for a meristem. And with the enough stochasticity, one of the sites will win over, and this will create your meristem. So one of the main goals of why we started creating this model was to try on some experiments. One of these experiments is called ablation experiments, where you're trying to cut out a certain part of your fern and try to see how this would affect the whole process and how would the fern react to that. So we're trying to hold experiments that could work or are enabled in both simulations and in the lab. So one type of these experiments that we looked at was ablating the meristem notch right after its creation. So as you can see in this picture, here we had a meristem and then we cut it out. So the results in both the simulations and in the lab showed that this system was able to recreate or reinitiate another meristem on the opposite side. So it really indicated a very cool, generative regeneration property for this system. So right now, what we're currently doing is we're trying to do more ablation experiments to figure out how would that affect different properties or roles of the fern. But what I can really say from what I have seen so far is that this system is really good at surviving. So almost always, no matter what you do, it finds its way to heal and then survive. And I think this is where we're at a phase where we're looking at the model and at the lab. So we're at the phase of interaction between math and biology, where the math is helping us understand the biology and the biology is helping us fine-tune our model. So I think this is one of the highlights of this workshop and one of the main goals of NITMB. So I think this system is really interesting in terms of wound healing, and I think it would have a really interesting future if we really looked into it further. So I really wanted to share that with you today.
I
think it's very exciting. I'm in neuroscience, but this is something very similar what we see in human or other animal embryos too, where you can kind of take a part of their notochord and they regenerate as like a different spinal axis and stuff like that. But then we see in embryos that at different developmental stages we can kind of lose the ability to do that as well. So it'd be curious to see at like what temporal scales does the regenerative properties of the meristem kind of change as the cycles proceed, right?
What we have seen so far, both in simulation and in the actual lab experiments, is that it's true that it is like affected with the time point at which you do the ablation. The closer you are to early stages, the more it's unlikely to survive, of course. And you can imagine like you're trying to ablate something that's less than 100 by 100 micrometers under the microscope. So you're very likely going to destroy it if you're not an expert. I've tried that and I did destroy it very badly.
Yeah, I don't know how much.
Go ahead.
Oh, I was just saying, I don't know how much we know about like the different transcription factors kind of regulating this process, but you could also kind of do like something CRISPR based where you can just inhibit those factors and kind of see how developmentally they arrest in terms of.
That's something that we're planning to like look into in the future. Yeah.
So here, like if I saw correctly the equation and remembered it correctly, which is always a hard thing for me apparently, but basically you are doing what I love, which is minimal modeling, and you're basically just looking at like the propagation and like diffusion of like some chemical signal, as opposed to this being some sort of like there's no like predetermined like genetic encoding that these cells are going to become this or that, right?
This is just like a very simple model.
What do you know? Like, did the biologists know like what that signal is?
Yeah, it's called like an auxin signal, and they had like markers to trace that and see how it like dissipates on the fern. They speculated that it's the only signal coming from the spore coat, but in our model we assumed we have like two signals.
So it's like two competing signals. Yeah. So I said, okay, cool.
Does your analysis help teach you why there are not more than one meristem generally per embryo? Like if you were able to make two meristems happen with an ablation experiment, like what control feature allows typical ferns to only have one?
So our collaborators have seen like very rare cases where you have two meristems initiating on both sides, but there were like very rare cases. In our model, we're using like randomness so that you can pick like the one that reaches a certain target of overlapping of intensity of the signals, so that you can choose which side. But I think that's something that we could add, like cases where you have two meristems.
Would you need like another signal to give you another meristem?
So our current model assumes that once you choose your meristem, it would start sending another signal that would inhibit everyone and prevent you from having another one on the other side.
So during this process, do cells become stem cells again and then turn into the other type?
Yeah, so around this area, it's called meristem because, yeah, this is like a type of stem cells.
Is this you doing an experiment also?
This is me trying to
That's really cool.
trying to ablate this thing, but I failed. It's really hard, and it made me really appreciate people working in the lab because it's really hard. Yes.
When you think of plants, you also think of circadian things, light, temperature, all these environmental cues.
Yeah, of course.
If these are also related to like when the regeneration is at its highest.
I think our collaborators do choose like a certain temperature where they carry their experiments. I know they also like change like certain circumstances by adding certain chemicals to the fern to block certain things. I'm really not aware of like the ranges of the temperatures used.
You could try it in the dark.
Yes. Yeah.
The dedifferentiation between the found meristem and the other cells very sharp?
Can you repeat the question?
Not really.
Can you repeat the question, please?
So is there like a very sharp boundary between the meristem part and the non-meristem part? I don't think we see that. So I think this is like the intensity of a certain, I think it's called DR5 marker or signal for the auxin signal. And I like you can see from the intensity, it's not really a sharp boundary between meristem and non-meristem.
Amazing. Well, thank Nour.